Thank you for having me here at the WikiPathways Summit. I'm very excited and honored to be here. I'm Jalal Siddiqui, I think I was already introduced, and I'm a postdoc in the laboratory of Dr. Avi Matei at The Ohio State University in the Biomedical Informatics Department. And I will be discussing RAMP. It's an integrated database of metabolite and gene pathway annotations that has been developed by our laboratory. So the focus of our lab is metabolomics, one of the major focuses. And so one question you're going to ask is, what are metabolites? Metabolites are small molecules, less than 1,500 kilodaltons, that are the intermediates and the products of metabolism. And these can include building blocks such as simple carbohydrates, simple lipids, amino acids, and nucleotides. These building blocks of much larger macromolecules. Or they can also include hormones and also drugs and xenobiotics present in the body. And so these are required for the structure of cells, cell signaling, transcriptional regulation, and in some, all sorts of biological processes. Now, in the whole dogma of molecular biology, the whole dogma of information flow in molecular biology that goes from DNA to RNA to proteins, metabolites are the most downstream level. And because of this, they're seen as a representation of phenotype. And it's no wonder that several studies have used them it's no wonder that metabolites have been used as biomarkers of cancer and other types of diseases. But one thing to note is that this flow is never one-way linear, because it's already known that so many proteins or transcriptional factors can influence the way DNA is converted to RNA. Or the information from DNA is how DNA is transcribed to RNA. And likewise, we know metabolites can influence how DNA will also be, how DNA can be transcribed into RNA and so forth, other processes. So in reality, the disease the characteristics of any phenotype or disease depend upon a range of different omes, such as the genome, the proteome, and the metabolome, as well as biological pathways and cellular context, and the environment. And so if you want to develop a more complete view of all these, of what's going on in a disease and how we might treat it, it's best to integrate these different omics data sets. And so there's really two major ways one can actually integrate these different data sets. One is a numerical-based approach, where you'll take a biological data set. So let's say, as an example, what we do is gene expression and metabolomics. And you're going to see maybe how the abundances will change from maybe a disease state to a controlled state, or how the relationship of the gene and metabolite abundances will change with respect to one another in two different states. And the other is a pathway-based approach, where you'll take the metabolite or the gene data or any other type of biological data set, and you will combine it with already curated information of biological pathways and processes. And our laboratory has developed both an example of each of these approaches. We've developed IntLim, or integration through linear modeling, that captures the gene metabolite pairs whose correlation or association will depend upon phenotype. As an example, like phenotype can be tumor versus non-tumor, breast cancer versus maybe lung cancer, and so forth. And so what we'll identify is there let's say a gene metabolite correlation could be in tumor cells very positively correlated, but in non-tumor tissue it may have a negative correlation or a weak correlation, and vice versa. And we've also developed RAMP, or Relational Database of Metabolomics Pathways. And so this is an integrated database of gene and metabolite annotations. And so it is, in essence, a public and comprehensive pathway database focused on human metabolite and gene annotations. And this is going to be the focus of my talk. And so what you're going to ask is, why are we making this other tool? And so one thing is that Our laboratory is finding ways to, was trying to, and is still trying to find ways to develop improved pathway analyses methods. And so to do so, we were looking for a database containing gene and metabolite pathway annotations. So we couldn't find an off-the-shelf database that contained all these gene metabolite pathway annotations. So we decided to create one ourselves. So RAMP is an open source standalone database that can be queried. The MySQL file is available, as well as Python file code used to reproduce the database. And so this allows it to be easily queried and also combined with other analyses tools. It also contains up-to-date comprehensive annotations from WikiPathways, KEGG, Reactome, and HMDB. And furthermore, it is the basis for improving pathway analysis methods. One example which we'll show today is about removing pathway redundancy, which is our functional clustering tool, and also incorporating other knowledge. And we are also working on an improved a new pathway analysis methods that uses a Bayes approach. And so RAMP is composed of, integrates metabolite and gene expression data from WikiPathways, HMDB, KEGG, and Reactome. We have around 1,400 compounds from WikiPathways and around 6,000 unique genes from WikiPathways and also 411 pathways. With HMDB, which is the largest database of compounds in humans, we have over 100,000 metabolites, around 5,000, around 6,000 genes and 50,000 pathways. And from KEGG, we have around 3,000 to 4,000 metabolites, 7,000 genes, and over 300 pathways. And we also have similar amounts from Reactome, about 1,700 metabolites, 11,000 genes, and over 2,000 pathways. And so you're going to ask, why do you need to integrate all this data? So look here. So this is the overlap between the databases. So look, even though there is some overlap, it's actually really small. And these percentages are whatever is unique to that database. So if you look here, KEGG, 42% of what's in KEGG is only, of these metabolite annotations in KEGG are found only in KEGG. And likewise, you see a general pattern where about a quarter to a half of a lot of these annotations are only found in that particular database. And you can see like a quarter of the metabolites found in WikiPathways are only in WikiPathways. About a third of the genes in WikiPathways are also found only in WikiPathways. And you see this similar trend. Of course, like one exception might be HMDB, where 98% of the compounds are only found in HMDB, and the genes in KEGG, the human genes in KEGG, which would be around only 8.9% are only exclusive. But the whole take-home message is, even though we have 4 major databases, a lot of them contain very unique information, and so there is a good need to have, so there's no one be-all, end-all database, and so there is a good need to integrate. And so not only have we developed a database, but we've developed an R package that contains a user-friendly, shiny web interface that allows even non-computational users to be able to make queries. So this is just an example of our shiny app. We also have a web server which allows you to directly access the app without a need to download. So here, this front page will contain information on RAMP, and on the left side has the queries that that RAMP allows for, and I'll go over these right now. So we have 4 major queries using RAMP. So you can retrieve analytes given a user input pathway. So if I enter a pathway name, it will give you the analytes. Analytes meaning genes or metabolites that are contained within that pathway. You also can retrieve ontologies if you give a metabolite. So if I give a metabolite, I'll get the ontologies. An ontology for a metabolite will be, what type of biofluid is it? Is it in breast milk? Is it in urine? Is it in blood? Is it endogenous to humans, or is it something exogenous? Is where in the cell is it found? What type of tissues it's found in? This is the ontologies for these metabolites. We also find out analytes in the same reaction as user input analytes. So if you enter the name of a compound, you will find genes whose protein products are involved in reactions with that compound. If you enter a gene, you'll find metabolites which are involved in reactions with that with the gene's product. And this is kind of interesting because you can also develop networks, a network level visualization. Like we show here for glutamine and methionine, you see the proteins which interact with glutamine and methionine, and also which proteins are commonly interacting with both of them. And also we allow you to retrieve pathways. The other query we have is you can get pathways given user analytes. You enter the list of genes and/or metabolites, and you can get pathways that those genes and metabolites are annotated to. And this is the basis of our pathway enrichment analysis and also our functional clustering algorithms. Now, we already have implemented on the R package and our Shiny app functional clustering of pathways on a pathway enrichment analysis. And so we find which pathways are overrepresented in a given list of genes or metabolites. And so that will be done through Fisher's t-test for the gene list and the metabolite list. But we also allow the combining of the p-values for both genes and metabolites to get a combined p-value. So this allows for an integrated gene and metabolite pathway analysis. And also we have a tool known as functional clustering. So we will cluster, so even though with the pathway overrepresentation analysis, you'll get a list of enriched pathways based off your thresholds. But we also use the Jacquard index to find overlaps. And the reason is a lot of the pathways have contained overlapping information. And this is so, number one, for many reasons. One is you're grabbing pathways from so many different databases, from KEGG, Reactome, and WikiPathways. And so there's bound to be a lot of overlapping content. And furthermore, some of the pathways, like especially in Reactome, are hierarchical in nature. And so to reduce this redundancy, we have implemented this pathway clustering. So this will sort of reduce your list of enriched or overrepresented pathways into clusters of pathways, and that will ease interpretability. And so an example from some breast cancer pathway results, we're able to find pathways related to gene transcription and gene expression, and we're able to find all these pathways clustering in one group. And so I want to do one example query, which just to show an example of the app. So this allows you to retrieve analytes given a user input pathway. So this is one screen here. So this would this is our screen, our part of the screen of our app that allows you to query pathways, to query the pathways for analytes. So here you can just type in a pathway. Here we're interested in glucose metabolism. As you type in a pathway, you'll have a drop-down box which will give you the name of a corresponding pathway that's found in our database. And you also can choose whether you want to just have the compounds, the genes, or both. And so once you just click submit, it will say whether it's successful, and it will let you know what's the summary of results. And by generating a report, you can get the analytes that are present in the gene or metabolites present in that pathway of interest. And so just if you go down, you'll see all the analytes that you're interested in. So we're interested in both genes and metabolites. And so you get this table here that gives you a list, and this is the top 10 values of the analytes present in glucose metabolism. So, and one more thing is you don't have to just necessarily you don't have to just input one pathway. You can also input a list of pathways. And so this allows for batch queries as well. And so we have our R package and user-friendly Shiny app. So the R package, the MySQL dump files and code to reproduce the database are available on our GitHub repository. And we also have you're also able to access the Shiny app at the web server link right over here. And before I end, I'm just going to give a few acknowledgments. I would like to acknowledge the Matei Lab, my advisor, Dr. Evie Matei, who's right here, and Liz, Bofei, and Sen Yang were the top 3 first authors on this paper, and they've all moved on to their respective positions. Andy played a great role in developing the Shiny app and our package, and Manju is currently the maintainer of the database. And I also want to thank the NIH and also Ohio State University for the funding.